Chapter 17, Screencast 2, the Endocrine System, Hormone Solubility. This screencast will focus primarily between one of the most important characteristics of hormones and how to distinguish them, because it really does pretty much determine almost everything related to hormones. And that is going to be whether or not they are water-soluble or lipid-soluble. Now, when we look at lipid-soluble hormones, it's important to remember that, once again, they are capable of diffusing across the lipid bilayer of the cell membrane, but since they are not water-soluble, they're going to require a protein carrier as they move in an aqueous environment. Now, when we look at carriers, they are going to be proteins that are made by the liver. And a couple things to keep in mind, that these carrier proteins are actually going to protect these hormones from early destruction, and also that's going to play a role with respect to them having a longer half-life. Now, the thing to remember here is that the connection or binding between a lipid-soluble hormone and a carrier protein is temporary. They're going to have the ability to attach and detach. And so the thing to keep in mind here is the majority of the hormone will be bound hormone, but only the hormone that is not bound to the protein carrier is capable of binding to the intracellular receptor. Now, when we look at water-soluble hormones, they travel freely through the blood, and some may actually use a carrier protein, but it is a very small number of proteins. Now, let's take a look at this particular diagram. This is a figure that is no longer in our textbook. It was in a previous version, but I think it really is an important figure. So let's take a look at the top panel first. And the top panel is focusing on water-soluble hormones and how they are transported within the blood. Now, keep in mind, because we're talking about water-soluble hormones, they can actually be stored within the cell. They are not lipid soluble, so they are not going to move from high concentration to low concentration when there is a gradient. So they can be stored. Notice they are going to be released within the bloodstream, and as they are water soluble, they are not going to require a carrier protein. But because they don't have a carrier protein, this may contribute to a shorter half-life than what is commonly seen in lipid-soluble hormones. Then they eventually make their way to the target, and note here where the receptor is. The receptor is actually on the cell membrane, and that also should make sense once again, as because we are talking about a water-soluble hormone, it does not have the ability to cross the cell membrane, so it will bind to a receptor that is found on the membrane of the target. Now, when we look at lipid-soluble hormones, it really is best to think of them as opposites to each other. And what we're going to notice here is you cannot store them. And the reason why is, as soon as you have a concentration gradient, they're going to move from an area of high concentration to low concentration. And as they are lipid soluble, that will result in them leaving the cell. Now, when they are released into the bloodstream, because they are not water soluble, they're going to require a protein carrier. The majority of lipid-soluble hormones will be bound to a protein carrier, and it's not until it is released that it's actually capable of being active. And once it is released, it will actually bind itself to the receptor, which we are going to find inside of the cell overall. Now, when we look at what is responsible for determining the overall level of a circulating hormone, it's going to be the relationship between two factors. Those two factors are going to be the rate at which a hormone is released, and so the more of a hormone that's going to be released, there is going to be a higher concentration of that. Now, the other aspect is going to be the elimination of that hormone. And this can occur a number of different ways. Typically, many hormones are going to be broken down within the liver through enzymatic pathways. Some of them are going to be removed from the blood via the kidneys or target cell uptake. Now, one thing to keep in mind here, the faster the elimination rate, the lower the blood concentration, and vice versa. So kind of imagine if there is a problem with the liver, there may actually be an increase in the circulating level of that hormone because its primary source or type of how it's going to be excreted or broken down is through enzymatic activity within the liver. So make sure that you're comfortable with understanding that relationship. Now, as addressed previously, the half-life of a lipid-soluble hormone is going to be significantly greater than that of a water-soluble hormone. Now, when we talk about half-life, really what we're addressing is the amount of time that is necessary to reduce a hormone's concentration to half of its original level. And this will depend on efficiency of elimination. We know that hormones with a short half-life must be secreted much more frequently so that you can maintain a normal circulating concentration of that hormone. So water-soluble hormones typically have a much shorter half-life, and this accounts for the majority of our small peptide hormones, where the half-life is actually measured in minutes. Now, steroid hormones, on the other hand, will typically have a much longer half-life. Part of this is due to the fact that they are protected by carrier proteins as they move in the bloodstream. A really good example of a hormone that has a relatively long half-life is testosterone. And the half-life of testosterone is actually close to 12 days. Now, here we're basically looking at the general mechanism in which a lipid-soluble hormone is going to work. Now, the next slide is going to be an animation, and I'm not going to go through that animation. You have the ability to go through that on your own. But I do want to walk through this particular image. So the first thing that's going to happen, the hormone must be released from the protein carrier. Now, because it is lipid soluble, it actually will diffuse into the cell. Now, the receptor, which we'll refer to as an intracellular receptor, can be found either in the cytosol or more generally actually found within the nucleus of the cell. Now, once that hormone binds to the intracellular receptor, it's going to form what we refer to as a hormone receptor complex. Now, the hormone receptor complex binds with a specific DNA sequence, and that is referred to as the hormone response element, or HRE. The binding of the hormone receptor complex to the hormone response element will actually stimulate the synthesis of new messenger RNA. Now, that messenger RNA will actually exit the nucleus, and it's going to be translated by a ribosome into some new protein. So this is the general mechanism in how all lipid-soluble hormones are going to work. They act by generating or creating new proteins. This is one of the reasons why it's going to take a while for them to actually exert their impact. So basically, your cell is retooling the factory to actually produce a new product. And that's just not going to happen right away. Now, feel free to take a listen or look at this particular animation, and it's going to walk you through visually the process of how lipid-soluble hormones actually work. Now we have to talk about water-soluble hormones. Now remember, these are actually going to use membrane receptors. And remember, hormones that are water-soluble are polar, so they do not have the ability to diffuse through the cell membrane. They're going to act through what we refer to as a signal transduction pathway. In this scenario, the hormone will act as the first messenger. It will initiate changes in cell activity by binding to the receptor. Now, keep in mind, it is ultimately the receptor, once activated, that is determining any change in cellular activity. Probably one of the most common mechanisms or variation on a theme, as I'm going to refer to it, is the activation of a G protein. This is simply an internal membrane protein that's going to bind a guanine nucleotide, hence G protein. And what we'll focus on is the activation of this G protein. Now, typically, once a G protein is activated, we're actually going to see some type of a enzymatic cascade. That's really what's going to happen. There may actually be the activation of a second messenger, and this could be a substance that will further monitor or modulate activity within that cell. So let's take a look at this variation on a theme per se. Now, what our textbook does not demonstrate is sometimes you may have a receptor that is bound to an ion channel. And once that hormone binds to the receptor, the impact on the ion channel is to either open the channel or close the channel. So this will happen very quickly, and what will end up happening is a change in the electrical properties of that particular cell. But let's look at what's happening once a G protein is activated. So here we can see that there is an inactive G protein, and once the chemical messenger or hormone, known as the first messenger, binds to the receptor, we are now going to notice that this G protein is actually activated. You don't need to know the actual process, just the fact that it is now activated. Now, once this G protein is activated, it's going to be released from the receptor, and it's going to move along the inside of the plasma membrane, and this is going to result in the formation or availability of a second messenger. Here is a different example of a G protein chemical messenger, and it's going to be glucagon. And glucagon is a hormone that's actually going to increase blood glucose levels. So once the activated G protein binds to and causes activation of the plasma membrane enzyme adenylate cyclase, so here an enzyme is activated in this particular situation, and it is actually going to result in creating something known as cyclic AMP. This is a very potent second messenger. It is one of the more common second messenger mechanisms that we have within the body. And what that is going to do, it's actually going to activate a protein kinase cascade. And what that does, these proteins are going to be phosphorylated, and that generally is going to activate those proteins. So the other thing to remember here is when we talk about water-soluble proteins, they are going to be activating proteins that are already available. They are not synthesizing new proteins. That is actually the mechanism of lipid-soluble hormones. Now here we're looking at epinephrine. And of course, it looks a little bit more complicated for the most part, but the common theme is the same. So it's a variation of a theme. We're actually going to see a G protein that has been activated when the hormone binds to the receptor. We're eventually going to get into second messengers. Here, this is different because we're talking about multiple second messengers. But the key thing that we're going to be looking at here is the inflow of calcium. So in response to this hormone, a calcium channel is going to be opened, and calcium is actually going to move into the cell. It is going to bind to a substance known as calmodulin, and this eventually is what's going to result in the activation of that kinase cascade. Now, notice I've been pretty generic about how they work. Now, let's take a step back, and we need to remember that there are lots of water-soluble hormones. So because of that, they do have a general mechanism which they are going to follow, hence the saying that variation on a theme. But what they end up causing to happen are lots of different things. Examples of these would be the activation or inhibition of an enzymatic pathway. It may promote growth or activating cellular division. It could cause the release of cellular secretions. We might see the contraction or relaxation of muscles. And we already kind of addressed a change in membrane permeability, which potentially can change the electrical properties of a given cell. Here, this is going to be a short video clip, and it's going to walk you through the general concept of how a water-soluble hormone is going to work. It will walk you through the activation of the G protein. And keep in mind, you're responsible for the general process or the theme that we have addressed here. You are not responsible for the alphabet soup and the other things that are actually addressed in this particular video. Now, as we've talked about before, generally what we're going to see is an intracellular enzymatic cascade. And what this means here is that a signal is amplified at each enzymatic step. So what this means is just a few molecules of a hormone can significantly change many molecules within the cell. Now, there are some places where this pathway can actually be regulated, and there are going to be some controls here. And cells actually possess mechanisms to quickly inactivate intermediates. One example would be cells having the ability to break down second messengers. And remember, second messengers are going to be those substances that are eventually responsible for changes in cell activity or to further activate a kinase cascade. Now, the next concept that we're going to be looking at is upregulation versus downregulation. And what we're talking about is changing the sensitivity of a cell in response to the amount of a particular circulating level of a hormone. Now, this is important to remember here that the number of receptors on a cell is not static. It is going to be dynamic, and it's going to change in response to the amount of the hormone in circulation. Now, I'm going to move on to the next diagram, but the thing to remember here is increasing the number of receptors is increasing the sensitivity, whereas downregulation will be decreasing the sensitivity to the hormone. So let's kind of take a look at this. So first, we're going to talk about upregulation. And what upregulation is, this is when the target cell will increase the number of receptors in response to a lower amount of that hormone. And this kind of makes sense. If I have a lower circulating level, I can make up for that by having more receptors or targets for that hormone, making it more likely that there is going to be an interaction between the hormone and the target. The opposite of that is downregulation. So example, for some reason, there is an increased level of that particular hormone, and the cellular response is going to be a decrease in the number of receptors. Since there is a high circulating concentration of the hormone, this cell is not going to require as many receptors to result in some activation due to the hormone we are addressing. Now, one of the things we want to address is how hormones interact with each other. And we're going to use three terms here. We are actually going to use synergistic, permissive, and antagonistic. So when we look at synergistic, we're actually talking about hormones that are working together to actually produce a greater effect. Permissive, this is when the first hormone allows the action of a second hormone. The example that I can give you here is the relationship between prolactin and oxytocin. Prolactin is a hormone that's responsible for the production of milk, whereas oxytocin is ejection. So you must have the milk first before it can actually be ejected. So these hormones have to be present and active in the right order. The other is antagonistic. So one hormone will oppose the action of another. And the best example we can address here is going to be insulin and glucagon. Insulin is responsible for decreasing blood glucose, whereas glucagon will actually increase blood glucose.